Let's just jump right in. Welcome to today's Explainer, where we are looking at the absolute frontier of modern biology. I'm talking about turning our own immune systems into literal cancer-curing superweapons. We're stepping completely out of the era of blunt medical instruments and into the age of genomic engineering, where we can actually rewrite the source code of human survival. If you've ever found yourself wondering just how close we are to a real cure for cancer, well, the breakthroughs we're looking at today are absolutely going to blow your mind. To really get this, we got to start with a wild medical mystery. Let's rewind to 2012. Imagine a 7-year-old girl, Emily Whitehead. She was battling a highly aggressive leukemia that had just stubbornly resisted every single standard treatment available. Her parents were given literally the most devastating news imaginable. They were told to prepare for hospice. It was, for all intents and purposes, the end of the line for traditional medicine.
But instead of giving up, a team of pioneering scientists tried something that sounds like it was ripped straight out of a sci-fi movie. They actually extracted Emily's own immune cells right out of her blood, took them into a lab, and gave them an entirely new set of genetic instructions. Basically, they reprogrammed her biology to search and destroy her specific cancer.
And this right here perfectly captures the giant step function leap we've made in modern medicine. Because of those reprogrammed cells, Emily didn't just survive a terminal diagnosis. Today, she's a perfectly healthy, thriving college student. A terminal diagnosis completely overturned just by hacking her own cells. Incredible, right?
So, to understand exactly how her miraculous cure worked and what this means for the future of healthcare, here's our roadmap for today's Explainer. We're going to hit 4 main areas: the immune surveillance team, hacking cells with CRISPR, targeting solid tumors, and finally, actionable steps for lowering your cancer risk.
All right, Section 1, the immune surveillance team. Let's zoom way in and look at the microscopic battlefield inside our own bodies.
Now, as you probably know, your immune system is this highly coordinated network of white blood cells permeating every single organ. But the real MVPs of this story, your T cells. Think of your T cells as an elite, highly trained surveillance team. Every single one has a specific sensor on its surface designed to recognize foreign invaders and take them out. And what's really cool is that these cells are actually educated in a specialized organ near your heart called the thymus. That's where they're carefully vetted to make sure they only attack actual threats, so they don't accidentally turn friendly fire on your own healthy tissue.
But here's the kicker. Cancer is fundamentally a genetic disease. It happens when our own healthy cells mutate and go rogue. Because those cancer cells look so much like our normal self, the immune system often hits the brakes. It just doesn't spot the danger, which is exactly why, for decades, we've had to rely on chemotherapy, which, let's be honest, is basically pumping the body full of toxins and crossing our fingers that the poison kills the cancer faster than it kills the healthy tissue. But CAR T-cell therapy, it's a total game changer. We're no longer relying on nature's faulty sensors. We are engineering precision-programmed super soldiers.
Which brings us perfectly into Section 2, hacking cells with CRISPR.
So how do we actually go about arming these super soldiers in the lab? Well, the protocol that saved Emily is called CAR T, but this whole field has been thrown into absolute hyperdrive by CRISPR-Cas9. You've probably heard of it. CRISPR is essentially a pair of highly programmable molecular scissors. And the craziest part? We didn't even invent it from scratch. It was discovered as a natural defense mechanism that bacteria use to literally chop up invading viruses. We basically just hijacked this bacterial defense system, and now we use it to rewrite the source code of human DNA.
And the most crucial takeaway here is just how mind-bogglingly simple this lab process has become. To edit a genome today, a scientist literally just orders a specific sequence of RNA off the internet. Yeah, off the internet. They take that RNA, mix it with the Cas9 molecular scissors, and drop it into human cells in a lab dish. Within a matter of days, the CRISPR system finds the exact target, makes a precise cut, and edits the human source code. It is just an unimaginably powerful tool.
And science is already iterating to make it even safer. We can now take it a step further using something called epi-editing. With epi-editing, instead of making a permanent physical slice in the DNA strands, which, carries some risk of unintended consequences, we can just flip genetic switches on or off. It's like dimming a light switch instead of rewiring the whole house. We change how the cell behaves without permanently cutting its core structure, mitigating a massive amount of risk while still totally upgrading the cell's capabilities.
All right, let's keep moving to Section 3, targeting solid tumors, and see how we're taking this from blood cancers to much, much harder targets.
Curing leukemia in the bloodstream is one thing, but solid tumors like prostate cancer are a totally different beast. They create this harsh, localized, heavily immunosuppressive environment that actively fights off your immune cells. It's a fortress. But right now, companies like Arsenal Biosciences are running cutting-edge clinical trials to breach it. Get this, they're using CRISPR to write 10,000 letters of new code directly into a patient's T cells. 10,000! This massive software update doesn't just help the cells find the cancer, it literally gives them resilience genes to survive and keep fighting inside that toxic tumor environment.
Now, of course, as we push this genomic revolution forward, we really have to navigate the ethics of what we're doing. The big, crucial distinction here is between somatic edits and germline edits. All the life-saving work we're talking about today, those are somatic edits. That means genetic changes made strictly to a specific patient's cells to treat their current disease. It stops with them. That is totally different from germline edits, which would alter the DNA in a way that gets passed down to future generations, permanently altering human heredity. So to keep things safe and ethical, the medical field is keeping its focus strictly on treating the patient right in front of us.
Looking to the near future, though, things get even more wild. Right now, CAR-T is a heavy lift, taking cells out, editing them in a specialized lab, and putting them back. But soon, we might be banking our own healthy, youthful T cells when we're perfectly well, keeping them on ice just in case. And even more mind-blowing, scientists are developing tiny delivery vehicles called lipid nanoparticles. The ultimate goal is to literally inject these nanoparticles straight into your arm, and they deliver the gene edits to your T cells right inside your own body, bypassing the external lab entirely.
Finally, let's step out of the lab for Section 4, Lowering Your Cancer Risk. Let's talk about how you can be the hero of your own story. Because look, while these futuristic gene edits are incredibly thrilling, prevention is always the ultimate cure. As we age, our risk of cancer naturally goes up, simply because we've had more time to accumulate damage to our DNA. So you need to act as a scientist of one and build your armor by aggressively avoiding known mutagens, the stuff that actively breaks down your cell's source code. We're talking an absolute zero-tolerance policy for smoking and vaping, for sure, but it also means being smart about UV light exposure, maybe thinking twice before eating heavily charred meats every weekend, properly washing pesticides off your veggies, and just minimizing unnecessary low-level radiation. On top of that, being a scientist of one means really knowing your own baseline. If cancer runs in your family, you kind of need to know your specific genetic vulnerabilities. Testing for things like the BRCA gene mutation, which heavily predisposes people to breast and other cancers, is surprisingly affordable and widely available now. Actually, taking the initiative to know your status gives you a massive upper hand. It lets you team up with your doctor to put aggressive, tailored preventative measures in place way before disease ever gets a foothold.
Gene editing has truly crossed over from science fiction into an everyday clinical reality. It's giving us a level of control over our own biology that was literally unimaginable a decade ago. But I want to leave you with this provocative thought as we wrap up today's explainer. You hold tremendous power over your health right now, today. Take a close look at your daily environment, from the sun exposure you get to the food on your plate, and ask yourself, what is one mutagen you can permanently cut out of your routine this week to protect your biological source code? Take control of your biology, and I'll see you in the next explainer.